and then you just let me know when we think we're ready.
Yeah, I think you can start now. We usually have a few more people coming, but just let them filter in over the next few minutes, and get started.
Okay, cool. All right. Hey everyone, I'm Paige, a postdoc here at Macquarie University, working in the Centre of Excellence of Synthetic Biology under Ian Paulson, if you guys have heard of Ian before. And today I'm going to be talking about synthetic yeast chromosomes. And I guess I've been asked to do this because I have specifically worked on the Yeast 2.0 project, which I will talk more about throughout the lecture. No, I do just want to say today, if you guys have any questions at any point throughout the lecture, please pop your hand up, ask away, like think of it as more of a discussion because there is some complex engineering stuff that we're going to get into. So if that's confusing you, by all means, I'm more than happy to pause and have a chat about it.
Sure,
Um,
you guys can type into the chat as well, and I can
yeah,
let you know if you have any questions. We'll let Paige know along the way instead of at the end.
yeah. All right. So let's begin. So there we go. All right, so we're going to start with like the progression of how synthetic chromosomes came about. And this is kind of the timeline that we're going to go through. And it all kind of started about the mid 2000s, where we started to get the idea that we could create these synthetic yeasts. Um, and we'll go back even a little bit further. It's not on here, but in 1996, just as a reminder, we had the complete synthesis of, not synthesis, we had the complete sequencing of the yeast genome. And people started getting thinking, well, if we know what the genome looks like now, can we kind of create our own copy of it? And that's where it started to really come into. So, why would we actually want to create a synthetic chromosome, though, in the first place? Well, the first is because we can. We're scientists. We like to tinker with things and do some cool stuff. But it also means that we could have better control over the organism and do easier genetic manipulation of it. We could conduct large scale mutagenesis studies to help us get a better understanding of the organism, and also create these minimal genomes. So, a lot of organisms, they have a lot of DNA and a lot of stuff that it doesn't necessarily need, especially in industrial applications. And by reducing its genome to just contain, the things for survival and for it to produce what we want it to produce in an industrial setting, could be extremely beneficial for us. So that's just some of the reasons why we'd want to create a synthetic chromosome or a complete synthetic genome, which ultimately is the end goal. So the very first organism that was completely made synthetic was actually done in 2008, and it was a bacterium known as Mycoplasma genitalium. Now, this is the smallest genome that can independently be replicated in culture. And its entire genome is only 580,000 base pairs. To let you know, in yeast, that is, oops, sorry, my door is locked and I think someone is trying to get in. Give me 2 seconds. It's just the cleaners. um, so it is the smallest genome that can be replicated. It has a tiny complete genome of only 580,000 base pairs. And as I said, to put it into context, a yeast chromosome, one of their chromosomes can be larger than this. So it was kind of the easiest one to start with. In total, it has 485 protein coding genes, and only 100 of those were with unknown function. And so they decided to create this synthetic genome so they could get an understanding of which of these 100 genes were dispensable, what could they get rid of, did the cell not really need them, and so forth. And then in 2010, we had the second synthetic organism created, and that was Mycoplasma mycoides. I can never say these properly. And one of the reasons that this was of interest is because it infects cattle and goats, which obviously plays a big role in agriculture. So trying to get it to understand it a little bit better plays a key role there. All right. So before we actually got to creating a complete synthetic chromosome, it actually started out with a process known as scramble. So scramble stands for synthetic chromosome recombination and modification by LOXP mediated evolution. which is a little bit of a mouthful. And scramble was first designed in yeast in the chromosome 9 and chromosome 6. And it was only a partial bit of chromosome 6 that they did it. And so what they wanted to do was to see if they could like scramble up the genome and see if it would make the cell fitter. Did it do any other cool functions or anything like that? And when it came to designing this, they thought, all right, this should have near wild type fitness. And I'll explain what scramble actually does or what it means by LOXP. So LOXP is, I think it's like 36 base pairs that is found in the genome and is recognized by Cre recombinase. And when this Cre recombinase recognizes this LOXP sequence, it essentially chops it in half and creates a double-stranded break in the DNA. And then the cell kind of like freaks out, so it's got to like repair itself back together. But by synthetically inserting these all throughout a genome, you can scramble up the genome. But what you want to make sure is that when you insert those sequences, you don't randomly put it in an essential gene and then pretty much kill your cell, because that's not going to be very helpful. So one of the very first things that they thought of when designing chromosome 9 and a little bit of chromosome 6 is that it should have near wild type fitness. And the other things that they also looked at is it should have been lacking destabilizing elements such as tRNAs and transposase. So transposase or tRNAs, they usually have a lot of repeat regions in them. And when we get a lot of repeat regions, especially in yeast, it has homologous recombination and kind of recombines on itself, or the transposons move around the genome. So we get this a little bit of stability. And when you're doing something like scramble and you're mixing up the genomes, you want to make sure that what you're seeing or the results you see are coming from the scramble and not because the yeast has just decided to activate one of its transposons and move itself around. And they also wanted genetic flexibility. All right, so here's a visual of the types of things that can happen when you actually scramble up the genome. So you've got these LOXP sites, they've been put throughout chromosome 9 or they've been put out through chromosome 6. And we put in the Cre recombinase. So this is the enzyme that goes and recognizes those LOXP sites and cuts them up. So you can see here that we have gene 1, gene 2, gene 3, they're flanked by these little LOXP sites, and we activate Cre recombinase and we could get inversions, so complete genes flipping around. We could get translocation. So this is where an entire gene, or it could be 4 or 5 genes, it could be a complete loci, completely moves to maybe somewhere else on the same chromosome, or it gets moved onto another chromosome. We can get deletions, and maybe the cell has decided, hey, these genes aren't that important to me, I'm going to get rid of them. And that's, you know, the essence of evolution, where you either get more genes or you get rid of them if you don't need them. We can get duplication. So the cell's gone, oh, actually, this is a bit of a stressful situation. I find that having this gene over here is helping me survive more. Let's make a second copy of it and duplicate it. And what they did in these papers is that they activated scramble and they looked for the number of events of each one of these types of things happening. So an inversion, a deletion, a duplication, or a translocation. So down the bottom here, you can see that we've got the number of events and how many strains that this thing was occurring in. And you can see here that deletions actually happened quite significantly in a lot of strains, which is kind of expected because, as I said, we want to create minimal genomes. Yeast has a lot of genes that it kind of actually doesn't really need. So it probably got rid of some of them. And so what's really cool about scramble is that you could have a normal population and you activate scramble and you get all this gene rearrangements occurring. And what happens in one single cell isn't going to be the same that happens in another single cell. And so you get this mixed population of all these different types of gene rearrangements, which means that they might have all these different types of phenotypes. So say, for instance, you did scramble and then wanted to see if it grew on alternative carbon sources such as ethanol. So yeast, by the way, likes to usually grow on sugar. So you could see if yeast grows on ethanol or methanol, and maybe the ones that have had the genome rearrangement, maybe a couple of them grow better on that. And so you would select those for those phenotypes, or you could look for something else. And what we've got here on the left is a graph showing the original chromosome length and the rearrangements that have occurred. So you can see here, this is kind of the controls of what they usually look like. And you can see here that this particular strain had so many deletions in it that it's nearly half the size. And then this particular strain, again, a lot of deletions, and you can see a lot of them actually had most of the deletions. And then these ones down here all show types of duplications that are occurring. And you can see that they've color coded them. So, you know, the purple here, this is in this bottom one, this purple loci is being replicated a lot. So this was the very first kind of example of creating a synthetic chromosome. Yeah, I can see something in the chat. Oh, Lindsay, they can't hear any sound.
I can hear sound. Are there other people who aren't able to hear the sound? Yeah. Yeah. Audio is fine for everyone else. So Tazim, I think it might be your computer or your system. Okay.
All right. We'll just keep going and hopefully they can fix up that issue. So Scramble, as I mentioned, was one of the very first examples of having a synthetic chromosome. And this is just one of the examples of what you can do with a synthetic chromosome and how we can create mixed populations that may have really strong phenotypes or something that we haven't seen before. So it's kind of like, think about it as fast forwarding evolution, which is kind of pretty cool. All right. There we go. All right. So once they did Scramble and they saw that it had worked and they partially created these synthetic chromosomes, they were like, whoa, okay, let's create a completely synthetic one and let's start with chromosome 3. And they did this and they published it in 2014. So a couple of years after the Scramble came out in 2011. Now, the reason they chose chromosome 3 is because it is the smallest chromosome in the 16 of yeast. So obviously being the smallest, kind of the easiest to do it. And as I mentioned before, when they were creating this in the Scramble experiment, there was a couple of features of the chromosomes that they wanted to keep. So it still had wild type fitness, that they had transposons removed. And so when they decided to design Syn3, as we would call it, there were several features that they wanted to put into a synthetic chromosome. And this kind of became like the Bible for every other single chromosome that eventually got built. So what are these differences and why did they do them? So on the left here, we've got a wild type strain, and on the right, we have a synthetic strain. And so the first thing that they designed was, all right, we're going to create our own synthetic telomeres. And this just gave them a little bit more control when it came to the engineering side of things. They took out all of the tRNAs. So just as a reminder, tRNAs are what literally guide your amino acid when you're creating an amino acid chain. So they got rid of all of the tRNAs and they actually put them onto a synthetic tRNA. So this neochromosome is what we call them. So it's essentially a new chromosome that they've built, but it's circular. This is where all of the tRNAs that were present in Syn3 were put onto. They created a synthetic centromere similar to the synthetic telomeres so that they had a bit more control over it. They changed all of the stop codons to TAA. Now, there's been shown in other studies that organisms literally only have one stop codon that they use, even though there is the genetic code for 3 of them. And they thought by creating all of stop codons to be TAA, it would allow them to use the other 2 stop codons in future experiments. They removed all non-essential introns. Saccharomyces cerevisiae, our model strain doesn't actually have that many introns, so to remove the non-essential ones was quite easy. And again, that's just leaning towards that minimal genome vibe that they were going for. As I mentioned, they got rid of all of the retrotransposon, all the transposons. These are unstable elements, as I've mentioned, because they often have repeat regions, and these repeat regions can kind of move themselves around and stuff, which is not exactly what you want. And it's kind of taking control away from the researchers. And the other key thing that they did, now this is really important when it comes to the actual design of a synthetic chromosome, is the introduction of PCR tags. So they created PCR tags that would recognize a wild type sequence, and they created PCR tags that would recognize a synthetic sequence. And when you get to actually building a chromosome, distinguishing whether your technique has worked and you've introduced the synthetic DNA is really important. And they obviously also introduced all of those LOXP sites. And the LOXP, as I mentioned, is really important to induce scramble and to get that rearrangement of the genomes. So these are all of the features of a synthetic chromosome. It was first done in SYN3, and as I mentioned, it was the Bible that pretty much moved forward for all of the other chromosomes. Now, most of this work was originally done by Jeff Booker here, and they thought, all right, well, we've got our first synthetic yeast chromosome. Can we do all of the other ones? Now, as you can see, from 2011 to 2014, that's 3 years. It's quite a big task to just create one synthetic chromosome. As I mentioned, yeast has 16 chromosomes. So they were like, okay, we as a lab can't do all of them. Let's create a worldwide project and kind of delegate all the other chromosomes to participating labs around the world. So we've got Leslie Mitchell, who was a employee within Jeff Booker's lab, created a program to go through each of the synthetic chromosomes and say, all right, these are all the stop codons that need to be changed. This is where we're putting the LOXP sites in. These are the introns that are removed, and pretty much all of those features of a synthetic chromosome that I've mentioned. They created it and said, all right, here's where all of the chromosomes are going to go, and these are all the universities that participated in it. And so it got distributed pretty much around the world, which created a consortium. We called it the Yeast 2.0 Consortium. I cannot say that word. It was originally called Synthetic Yeast 2.0, but they changed it to Yeast 2.0 because I think it just rolls off the tongue a little bit better. And in 2015, that's when everything was assigned. All right, and what's really cool is that Macquarie University actually got 2 of the chromosomes. So chromosome 14 was led by Tom Williams, and I think maybe he used to give this lecture, or he used to give a few lectures back in the past. And then the other 2, the other chromosome, chromosome 16. All good there? No questions? Nope. All right, cool. The other chromosome was chromosome 16, and this was led in partnership with Macquarie University and DPI from New South Wales, which is the Department of Industry and Fisheries. And this was with Hugh Gould and Heinrich Kuhkamp. And here's just an overall view of also of like how all the different types of features of each of the chromosomes. So, you know, the number of stop codons that was swapped out, like 75,000. That's a lot. The overall size of the synthetic version of the chromosome, you know, PCR tags and all of that. And you can actually see that chromosome 16 is one of the larger chromosomes, and it took 10 years to finish it, just to put into perspective of how big that is. And I will get into further details about why it took us 10 years. And and where I fit into this picture. I felt I was hired to work with Hugh to finish off chromosome 16 a few years ago. All right, so that's the overall picture of yeast 2.0. Do we have any questions so far about any of the stuff, especially the scramble stuff, which was a little bit confusing?
So Pfizer wants to know, is scramble a sort of CRISPR-Cas9? Can we use it like CRISPR on other organisms?
No. So it's not like CRISPR-Cas9. However, I will be talking about a CRISPR-Cas9 system a little bit further on how we use that in the creation or debugging of synthetic chromosomes, but no, scramble doesn't work like CRISPR. The only thing that I would say is that the only thing that makes it similar to CRISPR is that the Cre recombinase that is used literally cuts the DNA in a double-stranded break, which is what CRISPR would do. But you don't have any, so where you put the LOXP site, the Cre recombinase is going to cut there, but it doesn't, sometimes the efficiency can be a little bit off, right? So just because you've got 30 LOXP sites within the genome or within a chromosome, it's not going to cut all 30 of them, because you can also imagine that if you cut all 30 at once, you're going to butcher the cell and it's going to die instantly. It's not going to be able to repair that quickly. So we do find that when we do scramble, we are only getting maybe 1 or 2 events occurring, maybe a couple more. Does that help answer your question?
Okay, Nick is also has a question. Can scramble be used in other organisms or just in yeast because of their homologous recombination ability?
Good question. To be honest, I've only ever really seen it in yeast. I haven't really seen it elsewhere. But I can look that up for you and let you know. But yeah, I do think the big advantage of yeast is obviously because of its homologous recombination. And that's specifically even to baker's yeast. Like if we look at Euroia lipolytica, which is another industrial yeast strain, if we look at the fungal pathogen Cryptococcus neoformans, their homologous recombination is absolutely atrocious, and it does make engineering very difficult. So yeah. All right. Any others before I move on? Lindsay, you're on mute.
Oh yeah, no, I was saying, so the answer is maybe, but only maybe if they have some homologous recombination abilities.
Yeah, I know there is some bacteria that is kind of pretty good at it, but I don't think that they're not as like studied as much as like what Saccharomyces is. Alrighty, so we're about to get into how do you actually build a synthetic chromosome? All right, and that's just a little go Macquarie, because there wasn't too many other universities or groups around the world that actually got to do 2 of the chromosomes, which is exciting. All right, so how do we actually build a synthetic chromosome? So every single chromosome that there is out of all 16 of them, each and every single one of them was broken up into what we called megachunk. I don't know who came up with this naming system. It makes me laugh all the time. And megachunk is a joke that goes throughout the lab for the last 10 years. So each chromosome was broken up into pretty much the alphabet. We called these megachunks. We had one of the megachunks was literally just the centromere. And each of these megachunks were made up of chunks. And these chunks were also made up of mini chunks, but I'm not going to get into that detail. Now, each of these chunks were originally on a vector, PCR'd, amplified up, purified, and then co-transformed into a yeast. Now, what's really important is that, yes, chunky. What's really important to note is that each of these chunks had a tiny little bit of homology to each other. So where A1 is here, there was a tiny little bit of homology to this part of A2, and this part of A2 had a bit of homology to A3. And this just started to help stick everything together when it came to actually co-transforming it into yeast. And this is how it looked. So when you co-transformed it in, as I said, there was these homology bits. However, this part of A1 had homology to the genome locus. And what I should have also mentioned is that A4 had a selectable marker on it. So just as a reminder, selectable markers is where we can select to see if your genetic DNA or whatever you're putting in has actually gone in. In the case for the Yeast 2.0 project, the two markers used were URA3 and LU2. So these are auxotrophic markers, meaning that if we were to grow a yeast that is auxotrophic to ura on media that doesn't contain any ura, it won't be able to grow because it won't be able to synthesize its own ura for survival. If, however, we were to introduce the ura marker into the yeast and then grow it on media without uracil, it'll be able to grow fine because it's able to produce its own ura. And the same goes with Lu. We grow it on media that lacks leucine. Do we have any questions about selectable markers at all? Nope. Cool. So in this case, the ura marker gets put in and we have homology here to this part of the locus. And once we select that it's all gone in, this is now what our genome looks like. And we've successfully put in that A1 megachunk. And we confirm with this with PCR tags, and I'll show you how that looks in a little bit. So then we want to put in megachunk B. So what we've got here is we've got B1 actually has homology to that ura marker. And this time we're actually going to put in LU2 instead. And when we select for it, we're going to be growing it on LU2 plates, and we've removed that ura marker. So then we would use the ura marker for megachunk C, and so forth until we get the complete synthetic chromosome. Do we have any questions about that? And as we've already discussed, the only way this is really possible in Saccharomyces is because of its fantastic homologous recombination abilities. If we tried to do this in any other organism, it would be even more of a nightmare to what this was. Alrighty. So, as I mentioned, PCR tags were extremely important. And this was to help us distinguish that when we actually built it, did we actually get the synthetic DNA in, or do we still have the wild type DNA? So here we've got a wild type strain. We've screened it with these primers here, and we're getting amplification as expected, but we're not getting any amplification of the syn tag. So this indicates to us that we've got wild type in. However, when we look at the synthetic strain, we don't get any amplification of our wild type tags, but we get plenty of amplification of our synthetic tags. And that's what's really important. Now, we relied on these PCR tags a lot, especially when it came towards the end of building our chromosomes, because not everything works in a perfect world. And we found every now and again that while like most of the mega chunk went in, there was a tiny bit that stayed wild type, and it was like, okay, great. Now we've got to deal with that. Okay, so from 2015, when all the chromosomes were assigned, in 2017, we had the first kind of like cohort of chromosomes published in the science paper. And that's all these ones in blue. So they were completed and published. Now, what was really critical with kind of this negotiation that happened between science and the organizers of the Yeast 2.0 project is that they said, look, we will happily publish all of your chromosomes in our journal. However, you need to have kind of like a hook or an additional project on top of you saying, hey, I built a synthetic chromosome, because by the time you build one synthetic chromosome, the world's kind of like, it's been done. That's not exciting. So, you know, doing all the other chromosomes, you need to have something additional on top of it. So each group had to come up with also a unique project idea to add on to what they did. A lot of groups, you know, they initiated Scramble. I can't even remember some of the other stuff, but they had to do some extra things. And you'll also notice that a lot of the chromosomes that were published are these smaller ones here. A lot of the bigger ones, it took a lot longer. And the reason that they took a lot longer is that synthetic DNA can be unhealthy. As you can all imagine, you put in synthetic DNA, you're going to probably freak out the cell a little bit. And I'll also just let you know that when they designed all of the chromosomes back in 2015 or 2014, when they sent them all out to the groups, there was actually quite a lot of errors in what we call the in silico version. So what we mean by in silico is that we've got it on our computer screen. We're like, this is what we want to build. And then we found that there was a lot of errors in it, which isn't great. So we got all of these strains that were unhealthy and could not be published. And we had to go through a process known as debugging. All right. So this is what I mean by unhealthy, and this is how we could test for it. It's a simple spot assay. What we've got here is the synthetic strain, and what we've here is got wild type, and we can grow it on what we call optimal conditions. So this is YPD at 30 degrees. Now, yeast loves to grow at 30 degrees, and YPD is what we call a really nice, rich media. So it's happy, and it likes to grow. And you can see here that there's a little bit of a difference, but not much. They look pretty good. So we could say that it is wild type fitness in this case. However, if we were to grow it on YPD, so that's still nice rich media, but up it to 37 degrees, so this is a heat stress, you can see here that the synthetic strain actually doesn't grow that well compared to wild type. And this is a little bit of an issue because, as I mentioned, one of the criteria for our synthetic strains was that they had to have wild type fitness. And clearly right now, this does not have wild type fitness. Another stress that we can put it under, now this is a key one, is the mitochondria. So again, we're back at 30 degrees, and you can see that wild type actually doesn't grow as well as this one over here. And that's because we're growing on media known as YPG. So this is where we've swapped out the carbon source. So as I said, yeast loves to grow on sugar as its carbon source. So we've swapped out that carbon source to be a non-fermentable carbon source. And in this case, it is glycerol. And what happens is when you grow it on something like glycerol, you've really got to, it like activates its mitochondria, and you've got to really use that. So you put it under mitochondria stress. And you can see here that the synthetic strain just isn't performing as well as wild type. And you can imagine we can put it under full stress and it just does not grow. It is not good. That's a big sad. And essentially a lot of groups were finding that they created this synthetic chromosome and they were like, okay, we don't have wild type fitness. We need to go back to the drawing board, or we need to figure out where these bugs are essentially occurring in our synthetic chromosomes. So we can, the goal is to have a mostly completely synthetic chromosome, but if we have to create this one little gene and make it wild type and it's going to make it grow better, then that's fine. So we essentially, Macquarie particularly started on this thing called debugging, and this is where Cas9 came in. So debugging was introduced in 2022. So between 2017 and 2022, there was a lot of groups that were kind of just winging it and randomly finding these genes to be able to fix it. But Cas9, or this debugging approach, really helps accelerate how to fix this. So how does this work? So what we can do is we can get our synthetic chromosome, or our strain that has our specific synthetic chromosome in it, and we can mate it with a wild type copy of it. So all of our work is done in haploid cells. So this is one copy of the chromosomes, but here we're going to create a diploid. Now, inherently, diploids are just fitter than a haploid because it's got two copies of every single gene. And the theory is that most genes, if you have the better performing copy of it, is it's going to override kind of the bad one, and you're not going to really see a fitness defect. So we did find that when we mated our synthetic version with our wild type, that our strains did look a lot healthier compared to one with just the synthetic DNA. And this is though where we can use Cas9 to help us find the genes that are actually causing the defects. So what we did is we created a bunch of Cas9 vectors that had a PAM site that was specific to each and every single chunk. So we had our Cas9 that would target megachunk A, megachunk B, megachunk C, and so forth. And what it did is when it targeted, it would literally cut it at that point. And when you cut it, it kind of like gets rid of, so if we cut it at megachunk C, megachunk A, B, and C would like disappear, and the cell would be like, oh no, I'm missing an entire chromosome bit. But that's all right, it's good at homologous recombination, and it repairs it with the wild type strain. And what we found, or what you can find, is that your cell might actually start to grow sicker or grow better, and you can narrow down to where the DNA is turning synthetic and where it's turning wild type, and really narrow down those genes. So what I mean by that is we've cut it here and the cell is still growing happy. That's great. So we can say that probably this part of the genome, it's not really an issue. However, and same with this one here. However, if we have it happening a little bit further and our cells are starting to grow a bit sick, then we're like, hey, there's something interesting going on here. Let's take this specific strain, let's get it sequenced, let's see what it's looking like, and then we find, hey, it could be these genes here. What we do then is then we take those genes, we kind of do some reverse engineering to confirm that it's exactly that. We find it is exactly that, and then we fix it up in our synthetic chromosome that has one copy of its synthetic, so the haploid, and we slowly kept identifying genes, and we eventually got a synthetic chromosome that was a happy strain. And that is essentially how debugging worked. All right, so in 2013, so only last year, we have the second cohort of all of the synthetic yeast chromosomes published. This is what I mentioned by they had to put in a hook. So these guys used, I don't even know what chromosome that is, I think it's 13. They were looking at aging relating regulators. These guys looked at the effects of genome structure and function. They did a scramble on dysomic yeast. These guys did 3D organization and so forth. So lots of different things that could be done. So what does it mean in the future? What does this look like? So I can confirm now that all of the chromosomes have been synthetically made, and I believe that there is only two chromosomes that are left to be published completely, but I know that they're under review, and they should be getting comments back very, very soon. And I know every single group that had participated has sent all of their synthetic chromosomes back to Jeff Booker, who's based over at in New York. And it's some poor fella's job now. And I say poor because it took us 10 years to create a synthetic single chromosome. Some poor fella has to literally take every synthetic chromosome from each strain and put it all together to create a single synthetic complete genome. And it sounds like it's going to be a nightmare because we had a bunch of defects, another group had a bunch of defects, other groups had defects. And if you just imagine putting that all together in like one strain, you're going to have a very sick yeast. But that's not our problem to deal with. That is theirs. But yeah, so the future, are we going to see synthetic 3.0? I don't know what they're going to call it. You know, are we going to actually start seeing results of like evolution sped up where we're getting people doing all of this scramble and we're getting some really cool stuff? Like maybe we get a yeast that's like a super producer of biofuels or something. I don't know. Like there's some really cool stuff out there that can happen for it. So ending on that, these are pages of interest for you guys. Some of the stuff that just popped up in the lecture, and this is a shameless plug that if you want to listen to a synthetic talk, sorry, it's not a synthetic talk. If you want to listen to a comedy talk on synthetic yeast chromosomes, I presented it last year, and it's just a fun twist on that. So yeah,
We'll
guess
share
that's
that
it.
on our iLearn page for anyone who wants to
Yeah.
watch that later.
All right, I will stop sharing my screen, and actually no, I'll keep it up in case anyone has any questions, and I can go back to
Yeah.
any slides. So we've got
Thanks
some
for
time
that,
for some
Paige.
discussion.
We did hear from Briardo last week, so I think it was a good
Mhm.
carry on from what Briardo was talking about. He did introduce Scramble last week as well. And then in our lab yesterday, we were doing PCR and running gels. So they've just had an introduction to that as well. But most of
Oh,
them said they've
perfect.
already done PCR in the past, anyways, like previously. But yeah, do you guys have any questions for Paige on the last part of the talk or any part of the talk?
And also, I'm more than happy to ask, like, physical stuff in the lab, like what actually, like, I had to do and everything. Like, I ran probably 6 to 9 PCRs today. It was a lot. So
Yeah.
happy to answer any questions about that as well.
Well, I was telling them before how when I was doing like 8 plates a day that I developed like a big chicken drumstick thumb. So, has that happened to you from pipetting all the time? Or are you using a robot?
Um, I try to use the robots a lot now, which is very handy. But yes, I've definitely had like my thumb fatigue, um, doing a lot of pipetting. But what's really cool about, I guess, the center, is that we do have the robots and we can do high throughput studies now, which is super cool. And it definitely helped sped up this project. So when I was talking about the Cas9 stuff, when we were doing the debugging, we were screening, like, you cut with Cas9 at a particular mega chunk, and you would get like a plate with like hundreds of colonies. Now, where Cas9, when it cuts, even though it's cutting at the same site, so say for instance, we're cutting constantly at mega chunk D, when the cell repairs itself, it doesn't repair itself. And I probably should have mentioned this is pretty critical. It doesn't repair itself exactly the same way every time. So in one case where you've cut it, it might repair 5 kbs is still wild type and then it turns synthetic, or it might be in another one that it's 10 kbs where it's still wild type and then it's synthetic. So it's not identical every time, and that's where we can like narrow in and see the differences of it. So having the robots to like individually pick each cell to then run a PCR on it, to then do a growth assay, and growth assays are like, you've got to grow up a culture, you've got to plate it on those 4 different medias, you've got to do a serial dilution, you've got to spot each of those. So yeah, having the robots to do that, and literally we were screening every mega chunk for a defect. Yeah, definitely made things easier.
When you were doing this debugging, were there particular chunks that were really problematic? Like what do you think is the reason why certain chunks were chunkier than others and problematic?
So so um this was like a constant error that happened throughout the entire Yeast 2.0 project. And this is coming back to the original design that they did in silico in 2015. So when you look at a genome, any genome, but in particular yeast, you don't have genes going, all the same direction. You get them overlapping, and going in different directions. So one gene might go this way, another one might go this way, and then 2 of them might be going like this, and then they overlap just a little bit. Or you can have an annotated gene, which is like an actual gene that's doing a real function in the cell. And then you've got this like dubious small open reading frame that's like embedded in the opposite direction and like sits inside kind of like the open reading frame of this real gene. And what they've gone and done is they've inserted, this is key also, I didn't mention this, in where they inserted all of the LOXP sites in the genome, they inserted it at the end of every single gene in the genome. So if you can imagine that you've got this tiny small gene that's embedded in the opposite direction of this main gene, they've inserted a LOXP site. So they've just gone and disrupted the entire open reading frame of a functional gene that is essential for survival. And you're like, well, no wonder my cell's growing like crap. You've just gone and like inserted 36 base pairs into it. So we were finding that when we were doing the debugging, we were constantly finding LOXP sites inserted into a gene or even into the promoter region of a really important gene. And we found that if we then removed those LOXP sites, we were getting like increase fitness on YPG at 37 degrees, like tenfold.
Oh wow.
And it was like, why did no one go through and double check that we're putting in these LOXP sites into the right spot? So that was just like one. I wasn't part of the project back in 2014. I hadn't even started my PhD then. So, you know, I had no say in it. But the first time I saw everything, I was like, how does a bunch of scientists, like renowned scientists, miss something so critical? So one of the big chunks, there was a few chunks that were
Maybe
problematic.
they were men looking for something because they seem to have problems with finding things.
Oh, 100%. 100%.
Sorry,
So,
boy.
so the big chunks that were an issue were megachunk X, megachunk I, megachunk J, F, Y. So we had a couple.
Yeah.
So
That should be like
yeah,
all your new passwords now. It's just like the chunks that came from
the
each
chunks.
one.
Yeah, yeah.
No one
So
would have guessed that.
yeah. So yeah, that's kind of yeah, they were the trickier ones. But as I said, the minute we fixed them up and introduced like the to be honest, we even kept some of it still synthetic, and we just got rid of the LOXP sites, and even some PCR tags that they were putting in, completely. They put in one PCR and it changed the entire coding sequence of an entire gene, and we were like, how does your program? You're meant to just put in a PCR tag, but you've changed the entire coding sequence of an important gene.
Maybe their computers just weren't good enough back then to
No, I
process.
think so. I mean, I feel like 2014 was pretty ancient now when it
Yeah.
comes to computer technology.
I thought you were just saying that in general. I was going to say, hey, that's after I finished my PhD, so don't make me feel so aged. um, so I also was wondering about protein, like the translation and expression of proteins in these synthetic ones. So have you guys done anything like with proteomics to compare, you know, whether the proteins and glycosylation or those kind of things are like similar between the synthetic ones and the wild type ones?
Yeah, so we haven't looked at anything like glycosylation, but I actually did my PhD in proteomics, so it was quite handy. So we did do some whole cell proteomics on the synthetic strain and the wild type strain just to get a bit of an idea and a snapshot of, you know, what was going on. But we really didn't see anything significant or anything major. Like where we saw genes that were downregulated, it was mainly the mitochondria genes, which makes sense because it wasn't growing on mitochondria. But yeah, so we have done it, but we didn't really, and it was a very high level kind of analysis that we did. Like we could easily do a deep dive into what we detected, but just haven't done it.
Yeah, I just think it's interesting to find out, you know, whether the proteins are functioning the same ways or, you know, folded all the same ways or whatever, processed the same, stuff like that.
Yeah, I definitely was interested in it, but they were like, no, don't worry about it. We just need to get this finished. And I was like, okay.
Well, yeah, it sounds like something Nikki would be interested in. Nikki is my supervisor for like the past 8 years, so
Oh yeah, nice, nice, nice.
feel like she'd be interested at some point to do that.
Yeah, well, I think once we get it fully finished and off our backs, then I think there'll be like fresh energy coming into the project where we can actually do something with our chromosomes. Because right now, I think the big push has just been getting to finish them.
We just need to wait 10 more years until they assemble all of them together. But by that time, Nikki will probably be retired. So we'll have to be more.
Yeah, yeah, yeah.
Maybe even Ian will be retired. Who knows?
Oh, I think so. I think so. So does anyone have any other questions or like about anything? Like I'm more than happy to answer them. No. And I don't know if Lindsay just froze.
Yes, it just froze.
Okay.
Oh,
Okay.
back.
No, you're
All
back.
right. No one else is asking any questions. Do any of you guys have questions?
Also, I just want to say thank you all for coming. Like kudos for a late night lecture. There are so many of you.
Yeah, these are excellent students, Paige.
Yeah, I gave the class last year, and I think there was in person, and I think there was only like 8 or 9. So
Oh wow. Well, these
yeah,
are
this
these
is
are good kits. They also
really
made better gels yesterday than last year too. So I already gave them like a clap for that yesterday.
nice, nice. Making gels is key.
Yeah, their PCRs were better. So we also watched the Biorad GTCA song. Do you know this one? Oh my
No.
goodness. How do all you genomics people not do this? I'll send it to you after this.
Okay, all right, sounds good.
It's before your time because it would have been like 2014. So you were probably
Yeah.
in high school when it came out or something.
Yeah.
You'll love
All right,
it.
well,
It's about
all right.
genes.
Okay, nice. Well, if there's no questions, then I might just stop sharing my screen.
Yeah.
Um,
Great.
cool.
Thanks so much, Paige, for taking your time out on late at night, and we appreciate that. And I
No
hope everyone
worries.
has a great night. Say thanks to Paige, please. And hopefully you guys all have a good night too. Thanks, Paige.
Yes. Thank you.
Got lots of thanks rolling in for you.
Yes, I can see that all. Thank you all. All right, might see you guys around. You know, we're always keen to have people come through and check out the lab. So
Yeah.
keep that on your radar if any of that sounded really interesting to you guys.
You
'cause
should
we
show me around the lab. I actually haven't been in Unison Biolab, so
yes,
do
come
that.
come visit. Come visit. Because we
Okay.
do more than just synthetic yeast.
Yes. Yes.
All
Okay.
right.
Bye.
Bye.